Thank you, Alex, for that very generous introduction, and it's a pleasure to be here and tell you about the work that we've been doing at Insitro. And so Insitro fundamentally is a biology discovery company, although we make medicines at the end, and I'll talk a little bit about that. But the fundamental premise behind our work is that human biology is really complicated. Throughout my career, I've worked on multiple different challenging problems via machine learning, and biology is the most challenging by far. And I believe that the human brain will never be able to understand human biology. And so in order to get to something that translates to impactful medicines, we need to collect data at unprecedented scale across multiple levels of biology, and then let machines do what they currently do far better than people, which is interpret those data, find patterns, and help us redefine the complexity and heterogeneity of disease and identify effective intervention hubs. Data for us comes in two different forms. There's data that we print in our own wet lab environment, where we generate cellular models of disease, in vitro models of disease, and measure them using a range of unbiased ways of interrogating biology so that we don't come in with a preconception of what matters and what doesn't, but rather let the data speak for themselves. And then on the other side, because you can intervene in cells, but ultimately you want to intervene in human beings, we collect data from human patients. Again, focusing on high content, unbiased data modalities such as imaging and different types of omics, and of course human genetics, to give us an understanding of what health and disease looks like at the human level. We take those two types of data modalities and we integrate them via machine learning. So we've been doing, now I used to have to explain this notion of language learning, self-supervised learning. We've been doing that for 5 years. Now we don't need to explain that anymore because everyone's an expert on language learning. And so we basically do, what we do is learn the language of these biological data modalities. So not the language of text, but the language of histopathology, the language of cell morphology, the language of omics. We use that language learning to take the current fuzzy and imprecise definitions of what health and disease looks like and create something that is much more crisp and actionable. Action for us comes in the form of human genetics, because one of the things that is hardest in biology is to get at causality, and genetics is one of those few places where you have some assurance that an intervention is truly causal of the phenotypic changes that you're seeing. And so genetics for us comes in two forms. In our cellular systems, we intervene and actually edit the genome and measure the phenotypic consequences. And of course, in the human case, we use natural genetic changes, experiments of nature, like all of us in this room, to understand the genotype-phenotype causal relationship. And that together gives us a set of targets, which are high confidence genetic drivers of disease that we believe will meaningfully modulate clinical outcomes. Patients defined in a machine learning way via machine learning biomarkers that tell us in which patient an intervention is likely to make sense. And then we take that and we turn that into therapeutic interventions, which are molecules that modulate those targets in those patients. And that in turn will give us our pipeline of therapeutic interventions. So the combination of those two tools, what we call cellular machine learning and clinical machine learning, allow us to interrogate diseases across the spectrum of genetic architectures, and I'll show examples of those today. On the one side, you have diseases where there's a fair bit of genetic clarity. That is, you understand the genetic drivers of disease, they have reasonably high penetrance, and you can recreate those disease models in cellular systems by editing those disease-causing variants into isogenic lines, where the only difference is the disease-causing gene, and really try and understand in sort of a simulated case control setting what that disease does from a phenotypic perspective. On the other side, you have diseases where the genetic clarity is much more diffuse. The diseases are much more polygenic, and those are oftentimes also much more prevalent in the population, and so we can oftentimes rely on the phenotypic consequences in a large human population to understand what that genetic architecture does in a human organism. In the middle are diseases that have elements of both, where they are polygenic and diffuse, but at the same time there is a subset of patients where the disease is generated by high penetrance genetic drivers. And we like those diseases because it allows us to deploy our toolkit on both sides. And what we now are increasingly understanding, we as a community, is that many diseases, I would argue most diseases, are actually somewhere in the middle rather than the two extremes. Even diseases that have been typically viewed as Mendelian have a polygenic risk component that potentially exacerbates the disease. And even diseases that are typically viewed as polygenic and diffuse, diseases like diabetes or obesity, have high penetrance cases that help us understand the disease in this isogenic way. So we're deploying this platform that we're building across 3 therapeutic areas: metabolic disease, neuroscience, and oncology. What you see at the bottom is some of the machine learning-enabled microscopes that we've built for live cell imaging, which I won't get to talk about very much today. And what I'll show you today is a vignette in each of those 3 therapeutic areas. But first, let me tell you a little bit about our platform. So we really try, and as I mentioned earlier, create platforms that help us de-risk, and thanks to Alex for setting this up beautifully, the 3 different major phases of what it takes to bring medicines to patients. There's the biology discovery, figuring out what the right targets are in the right patients. There is the drug discovery, which accelerates the process of going from a target to a drug. And then at the end, there is the deployment of machine learning towards clinical strategy by using biomarkers to make our clinical trials more effective and efficient. So this is the platform that we're building. It involves the generation of very large amounts of data in both biology and chemistry, the acquisition of large amounts of clinical data across multiple modalities, all feeding into a unified data platform that is interrogated via a uniform set of algorithms, the same algorithms, whether you're doing histopath or cell morphology, and ultimately into our insight engine. And and then this is actually our mantra. This is our mission, which is we want to bring better drugs faster to the patients who can benefit most. That's where the biomarkers come in, through machine learning and data at scale, which we both generate and aggregate. And we think of this as the tagline of pipeline through platform, which is not something that is that prevalent in biotechnology, which is really generating a pipeline of medicines from an integrated, unified approach. Okay, so with that, I'm going to go into the vignettes, and the vignettes will illustrate that platform in action. So in oncology, we decided to focus on a foundational modality with a foundational language model where histopathology is that modality. The thing about histopathology is that it is probably the single most abundant modality in oncology. It is collected from pretty much every solid tumor patient almost anywhere in the world. It is increasingly digitized. And it is incredibly rich in its content. It has information all the way from subcellular structures through cell-to-cell interaction, all the way towards much coarser anatomical features such as vasculature. All of that richness, billions and billions of pixels per image, is summarized by pathologists typically into 3 qualitative numbers. So all of that richness is by and large getting lost in how these images are currently interrogated. And so what we've built is a foundational model for histopathology, which, as we'll show you, allows you to actually associate and impute multiple other high content modalities that give rise to novel targets. So we start with all of that information, all of those data. We apply a set of increasingly better self-supervised deep learning algorithms to generate an embedding. I used to have to explain that too, no longer. A lower dimension representation of those images. And with that low dimensional high content summarization, we can take much smaller data sets that have additional covariates, whether it's clinical covariates or genomic covariates, and be powered to find associations in ways that would not have been possible from looking at the raw histopathology on its own. And that allows us to identify new drivers of clinical outcomes. So first of all, this doesn't come for free. Better predictions really do start with better data. And so we started by looking at the histopath. We applied our self-supervised embedding. We got clusters that look like this, and like, what the heck is that black cluster? Turns out that black cluster is the ink markings on the images. So we applied an open source artifact removal platform didn't work really well. It actually removed the histo, left the markings, and so we ended up having to define our own machine learning informed pipeline for cleaning up histology images. With that, we tried to convince ourselves that there is indeed information left in these histology images that is currently going untapped. This is an illustration of that. What you see on the left is a standard Kaplan-Meier curve. For those of you who are not familiar, the x-axis is years from diagnosis. The y-axis is the fraction of patients that remain alive at that year. The curve, unfortunately, goes down to the right. And so what you see is the stratification between high-risk and low-risk patients via a model that is derived from the standard clinical covariates that are used in the standard of care, things like age, sex, tumor stage, and so on. On the right-hand side is what you get when you take the information from the embedding that was defined on the histopathology, the same one that the pathologist looks at, but with the embedding model, and you can see that this is a much cleaner stratification than you get from just the clinical covariates. So clearly there is information left sitting on the table that is not currently being deployed as part of the standard of care. Even more interesting than this slide is this slide, which shows the clinical risk score, which was used in the graph on the left, and the additional information derived from the embedding that was added on top of the clinical risk score. That's the y-axis. And what's notable is that correlation, and each dot here is a patient clinical risk score versus embedding risk score, correlation is 0. So that suggests that the information that the embedding is picking up on is actually entirely orthogonal to what the pathologist thinks to look for when grading these images, demonstrating just how much information is left over. Actually, I'm going to skip past this in the interest of time. So that's nice, but it doesn't actually give you novel targets. It's useful for biomarkers, it's useful for risk prediction or response prediction, but how do you get to targets? In order to get to actual molecular entities, we are overlaying those embeddings with additional molecular covariates. So here is almost like a proof of concept experiment where those molecular covariates are somatic alterations in the cancer genome. So cancer is a disease of genomic instability. Certain patients have disruptions, mutations in their genome, which oftentimes is a significant factor in the disease. And so the question is, can you actually predict those somatic alterations just by looking at the patient's histology sample and nothing else? And the answer is that you can, in fact, do that with incredibly high accuracy. A pathologist cannot do this in most cases at all. You can see accuracies of 90 to 95% looking at an image, figuring out what is the driver mutation within those images. Now, notably, there's other papers that did that at about the same time as we did this work, but they typically do that by having a special built neural network trained explicitly towards each of those alterations on its own with a very large number of parameters and a fairly large data set for that training problem. This is the same embedding that I showed you a couple of slides ago and a simple linear model on top of that, which again shows you just how much information there is in this language of histopathology. In order to communicate with pathologists and kind of explain what it is that the machine is picking up and also gain conviction ourselves that this is real, we developed a generative adversarial network approach for, you can think of it as the deep fake for pathology. So we start out with a tile that is obtained from these histopathology samples. And then we generate synthetic tiles which are moving in embedding space from CDH1 negative, say, to CDH1 positive. This is one of those cancer mutations. And basically generating synthetic histopath tiles where everything else is remaining equal except the status of that mutation. And we use that to communicate with pathologists and say, is what the machine is picking up on, does that make sense and does it teach you anything? And what you see on the right is the post hoc analysis by our pathologist who says, yep, I can see a clear histological progression from ductal to lobular carcinoma or whatever, so that you can really start to understand what the machine learning is identifying in these images. And this was done post hoc, wasn't input to the algorithm. So this is nice, but somatic alterations are very rarely novel targets because people know about them, they recognize them. If they were druggable, then there's already people working on that. How do you get to novel targets? So in order to do that, we ended up turning to a different modality that is a really cool assay that was developed at Stanford in about a decade ago by Howard Chang's lab. It's called ATAC-seq. ATAC-seq, for those of you who are not familiar with it, is a way of measuring whether the DNA, the chromatin, is open or closed at each region of the genome. And if a region of the genome is open, that is an indicator that whatever lies underneath that is likely active in this particular cell at this time. And so this is a cousin, if you will, to RNA sequencing, which also tells you which genes are expressed in a particular sample, but it has some benefits specifically, for example, in identifying lowly expressed genes, which are often harder to find in RNA-seq, as well as things that don't manifest in transcriptional activity, such as non-coding mutations and so on. Now, ATAC-seq is a really powerful assay, but it is also a quite finicky one, and specifically is quite sensitive to how samples are handled, and as such will likely not be used as part of the cancer standard of care. It's only by and large available in limited scale research data sets such as this one, which was a paper from also Howard Chang's lab, that was from 5 years ago, which looked at TCGA, the Cancer Genome Atlas, and picked 400 samples from within that and did bulk ATAC-seq on those 400 samples. So 400 is really not very many, especially when it's spread peanut butter thin across 23 cancer types. So the largest subcohort within that was about 30 breast cancer patients. You're not really powered for discovery with 30 patients. So the question that we asked ourselves is, can you, in the same way that we imputed somatic alterations, can you impute ATAC-seq? And when we first presented the idea to the people in the Chang lab who worked on this, they were like, "Yeah, you're gonna predict ATAC-seq from histology images. I mean, that's funny." But we decided to try, because if we would be successful in that, we would be able to take that data set that was generated on 400 patients and basically impute on, TCGA is 11,000 patients. At this point, we have close to 100,000 histo samples across multiple different cohorts from multiple different centers. So now we basically have we would be able to have 100,000 ATAC-seq. That would be pretty amazing, right? And so it turns out that you can, in fact, do that, not for every region in the genome, clearly, but for a good fraction. In this case, in the early work that we did, this is 1%. It's now north of that by a fair bit, where you can predict for those 1% of the regions of the genome, the ATAC-seq signature at an R² of greater than or equal to 0.5. And those are not just random regions of the genome, those are ones that manifest in histology and as such are likely to have an association with cancer. And by the way, when I say predict, I meant on held out test patients, needless to say. So what you see on the right is the actual ATAC-seq, the predicted ATAC-seq on held out patients, and then the bottom is a quantitative verification that similarity that you see is real. With this imputed ATAC-seq, we can now start to ask questions like the openness or closeness of which region of the genome is associated with survival. You couldn't do that with 30 patients, but now we have thousands of patients. And so you can start to create hazard ratios for dichotomized versions of ATAC-seq at different levels of activity of a particular gene. Do you see a stratification of survival for that particular patient? And you can see that you can get to hazard ratios that are actually quite interestingly significant, hazard ratios of 2 or so. And so with that, we were able to take, for example, triple-negative breast cancer, a disease that has very few meaningful treatments. I basically take all of the imputable ATAC-seq peaks and look at the gene that is underneath them and identify a bunch of genes, some of which are very well characterized, known TNBC targets. Those are the ones that are unblinded, as well as several new ones that have a hazard ratio of around 2 for TNBC patient survival. And while those have not previously, the new ones have not previously been implicated in TNBC, they have in most cases been implicated in breast cancer or other solid tumors. So these are potential new TNBC targets that we're currently validating in our labs. One form of validation that is not an experimental one, but a computational one, is we went back to TCGA, where there's, in addition to the attack-seq, there's also for each patient, the somatic alterations. And we asked whether those genes that we had identified as potential novel TNBC targets have an amplification event or any kind of somatic alteration event in those genes. And what you see here is the top 19. These are unfiltered. And the alteration events, each column is a patient, whether the patient had an alteration in that gene and what the nature of that alteration was. And you will see just by looking, at a high level that there is a proliferation of red, which corresponds to amplification events in those genes. That is a good indicator that the cancer likes to have that gene turned on or active. Now, there's also a statistical test for that we did not invent. It's called gain-loss ratio. And you can see just from that p-value that there is indeed statistical evidence that suggests that these genes do play a causal role in, the activation of those genes plays a causal role in tumor proliferation. Is this definitive evidence of causality? No, but it's certainly suggestive and helps encourage our conviction. So basically, this is what we've done in oncology, which is we onboard very high quality data, actually integrating multiple cohorts. We impute, and I talked about the imputation of attack-seq. I'll talk in a little bit about the imputation of RNA-seq, as well as other types of modalities that we're currently working on. That gives you a very rich, large cohort in which you can now start to look for associations between genetic, genomic, and clinical covariates, and that gives you a set of novel targets that you can then potentially explore as novel therapeutic interventions. Okay, moving on to our work in neuroscience, which is uses the other side of our work, which is the experimental platform that we've built in-house. And I will illustrate this using a close to monogenic disease called tuberous sclerosis complex. TSC, as it's called, is not something that many people know about, even though its prevalence is not that much lower than cystic fibrosis or other diseases that people are much more familiar with. It's a genetic sort of cancer. It's not really a cancer in the sense that it's proliferative but isn't metastatic. And so these, quote, benign tumors, as they're called, typically manifest in the CNS, the central nervous system. And when they do, those tumors generate refractory epilepsy. So for those of you who don't know what that means, it's 30 to 40 seizures a day in these pediatric patients. When you have 30 to 40 seizures a day, it pretty much fries your brain and gives rise to intellectual disability unless those seizures are kept under control. So how do we tackle this disease using our platform? So here we've built out this, as I said, extensive wet lab capability that takes cells, in this case and in many of our other work, induced pluripotent stem cells, and which are these cells that can turn into any lineage in the body if pushed in the right direction. And in this case, we took those cells, we introduced the disease-causing mutation, in this case TSC2, and so now you have what are called isogenic cells, which are identical, except for that one mutation that has been caused. We differentiated them into cortical neurons. We have state-of-the-art automated differentiation protocols that are done entirely by a robot. And then we measured those cells in a range of multimodal assays that range from fixed to live cell imaging, different types of omics, as well as you'll see electrophysiology. We learned an embedding on those cellular systems, and then on top of that embedding, we overlaid a disease axis where the positives for disease were the TSC knockouts, the negatives were the healthies, the wild-type cells, and that disease axis is now something that we can use to identify reversions of that disease phenotype. So first of all, we needed to convince ourselves that there was a disease phenotype and that it actually made sense, and so we spent a lot of time validating, or as we call it, credentialing phenotypes. So here is the RNA-seq phenotype. This is single-cell RNA-seq data that was obtained from these differentiated neurons. And on top of that, we overlaid the disease axis. And then we tested whether the disease axis corresponds to disease in a human. So how do you do that? an in vitro phenotype. One way is to take a treatment that you know has an effect. In this case, rapamycin is actually a valid therapeutic for this disease. It is known to be disease reverting. And so we put dose response of rapamycin into these cells, and you can see that it actually reverts along the disease axis. So it's a nice sort of conviction builder in the disease axis. Another experiment that we did is we took data from human patients. In this case, this is bulk RNA-seq from the brains of patients and the brains of healthy controls. That bulk RNA-seq was perfectly stratified using the model learned from the single-cell RNA-seq in vitro data. So again, is conviction that this is a meaningful disease axis that is relevant to human disease. We did the same kind of validation for other types of disease axes that we built, including this one, which uses live cell imaging, those pretty pictures that I showed, the pretty movies that I showed earlier, and that gave us a set of disease axes. So that's nice, but then how do you actually screen for reversions at scale? So one thing you could do is you could take each reversion that you want and put it in its own well and, you know, try and reconcile all of the batch effects and so on of each of those wells. We decided to go a different route that is harder, but we think has a lot of potential, which is pooled CRISPR screening. So in our case, we have a pool of cells, in this case, these NGN2 sick neurons, those TSC2 sick neurons, and we introduce a pool of CRISPR guides into those cells at a level that each cell, on average, gets a single guide. So now you have a pool of cells that are genetically heterogeneous in the sense that each cell has a different gene knocked out. And so now it's a pool of cells, and you can now go ahead and image those cells using our technology stack. You could do all sorts of things ranging from standard cell morphology, cell paint type stuff, specific localization of specific proteins, or live cell dynamics. That picture over here that you see is actually those little pink dots that are moving. Those are mitochondria moving from one axis, along axons from one neuron to the next. Having phenotyped each of those cells at a single cell resolution, we can now fix the cells and we sequence the barcode that defines the guide in each of those cells. So we built a sequencer inside the microscope. And so what you end up, and you can see the image that you see over there is the barcodes being sequenced one base pair at a time. That gives you a genotype-phenotype matrix at single cell resolution with full-blown morphological phenotypes for each of those cells. So that was the platform that we built. It was actually based on work that was done here at the Broad by Paul Blaney's lab. But we brought it to the next level because of the full morphological imaging. And that gave and then on top of that, we not only built it, we also productionized it. So this is an automated platform that has a robot that does all of those painful steps of, you know, the in situ sequencing and one base pair at a time and so on. And it requires a tremendous investment in chemistry, in automation, in data pipelines, and most notably in machine learning, because you can't even segment the cells properly without using machine learning to get to high quality of segmentation. So that gave us a screening platform, which allowed us to take what is currently, you know, a druggable genome scale, so about 5,000 guides. We're now actually bringing it up to full genome-wide screens and screen for reversions across the disease axis that I showed you earlier. And we ended up comparing both the transcriptional phenotyping and the image reversion, and they end up being correlated, but not overlapping entirely, and so it gave us complementary hits. And so we selected the 76 strongest hits across both of these for further validation. And validation for us has several different forms. I'm going to talk about two. One is support in human genetics. There is increasing realization that targets that have support in human genetics, that is, that they have an association with a human trait that is similar to the disease that you're trying to address, those are much more likely to be successful in the clinic. That's been published multiple times in multiple studies. So we look for associations with clinical traits that, for our targets, that are relevant, in this case, to tuberous sclerosis complex, and those phenotypes have to do with neurological disorders as well as proliferative disorders. And it turns out that we're very pleased that 55% of our targets, that's a very high hit rate, actually have genetic links to TSC relevant phenotypes. And most of those, you will notice, are in epilepsy and neurological disorders, and that is because our screening is done in neurons. Similar screens that were done in cancer cells, including across multiple labs and in our hands, did not give rise to that same enrichment for neurological phenotypes, and our conclusion is that modifiers are very cell type specific. So that is support in human genetics. The other form of evidence is an experimental assay that we built, which uses a multi-electrode array. So the neurons that we did our screen in are very young neurons. They're 10 days old. These neurons in the right-hand assay are grown to 50 days. And when you grow them out that long, they start talking to each other in the well. They start having network activity that is coordinated firing. And it is well known, as well as in our hands, that these TSC2 knockout neurons, like those of many other epilepsies, actually exhibit a hyperexcitability phenotype in the dish. That is, they start firing at very accelerated rates in certain periods in time in ways that are similar to the sort of intermittent hyperactivity that you see in patient EEGs in epilepsy. And so you can see that there is a very similar firing pattern. And so this is a highly translatable assay that gives you conviction would give you conviction in these targets. And so we took 14 of the hits that came out of the 76. We picked those not because they were necessarily the best, but because they had chemical tool compounds that allow us to do the experiment quickly. And this is the experiment for one of those targets, a novel target that you will not see on any mTOR pathway diagram. And what you see here is a almost perfect reversion of the hyperexcitability phenotype. So just to explain what you see here, on the left are two different hyperexcitability phenotypes, firing rates and burst frequency. The red is the sick neurons, the ones from the TSC knockout. You can see a much higher level of activity. The green bar that's next to it is the wild type, so very clearly different. The blue is the rapamycin reversion, so the positive control that we had, and the purple bars is a dose response for the small molecule tool compound that is associated with our target. And you can see a beautiful dose responsive reversion whose success rate is comparable to what you get from rapamycin, but from a completely novel target. We got this level of reversion from 50% of the 14 that we interrogated, including 6 that are entirely novel, which is, again, an unusually high hit rate. So those are currently we picked the favorites among those and we pushed those into drug discovery. I'm not going to talk a lot about our work in ALS today, partly because it's very much on progress. It's a wonderful partnership with BMS, and I don't need to explain, I think, about ALS. I think a lot of people are familiar with ALS, especially here. The thing about ALS is that it is one of those beautiful examples of things that are in the middle. There is 10% of ALS patients have familial mutations that are strong drivers of the disease. The remaining 90% are sporadics with much more diffuse genetics. The genetics are across multiple distinct pathways that range from mitochondrial dysfunction, autophagy, ER, and many others. And all of these eventually give rise to motor neuron death, but probably via somewhat different trajectories. And so in order to interrogate those different trajectories and find the right patient populations and the right modifiers for each patient population, we made a massive effort. We built the largest known library of isogenic iPSC lines where we engineered disease-causing mutations into those iPSC neurons across different genetic backgrounds. We generated robust, repeatable, automated differentiation processes into motor neurons, which are actually much harder to deal with in cortical neurons. The phenotyping that we talked about and the data pipelines. And as of earlier this year, we have pretty incredible ALS phenotypes and even some reversion hits, which I'm not going to be able to tell you about, but it's pretty exciting for us. So this is our CellML platform, which I illustrated in the context of neuroscience, human-derived biologically relevant cellular systems at unprecedented scale, multimodal phenotypes across both imaging and omic modalities, machine learning to identify cell states that align with human clinical outcomes, and then pooled genetic screening to identify novel targets that we then push into appropriate drug discovery efforts. Okay, I'm going to now talk about metabolic disease, which actually brings those two lines of work together, deploys both our clinical and cellular machine learning capabilities. This work started out actually in our partnership with Gilead in NASH, non-alcoholic steatohepatitis, which now has a different name, MASH, but whatever. And this was a data set that actually was what first developed our conviction in the amount of information in histopathology, because what we had here was close to 7,000 patients worth of liver biopsy data from 6 different Gilead clinical trials. A subset of those also had bulk RNA-seq, they had blood biomarkers, and an even smaller subset had genetics. And so here, we started out by looking at the histopathology data because NASH is actually only ascertained via histological features. That is the definition of the disease. And so Gilead had a single highly trained clinical pathologist who looked at every single sample, a single person, to avoid interpathologist variability. And we were like, okay, great, we're gonna look at genetic associations with these histology scores. And there was nothing. And the reason for that is that because, there's only 900 patients, and that's quite low powered for genetic associations. But the other thing that we realized is that it was very low resolution in terms of those phenotypes. They're very coarse-grained and a lot of variability, especially around boundaries. And so we basically deployed the same histopath embedding approach that I showed you earlier in the cancer only. We did this first. And we're able to sort of, again, demonstrate that there is a significant amount of information in those embeddings that is in those histopathology images that is currently going untapped. So here, for example, is the ability to predict gene expression in the liver, the bulk RNA-seq that were obtained from those biopsy samples from those embeddings, and you can see this is comparing to the pathologist scores, and every point that's above the diagonal on the left is a gene whose expression we can predict with much higher accuracy than you would from the pathology scores, and you can see that's pretty much all of them. And so again, the ability to predict molecular data that is much more now actionable from the histology images. This is actually one of my favorite slides because I think it really illustrates the two different sources of power that you get from machine learning interrogation of these quantitative phenotypes. So one of the things that we tried to do was associate with genetic variants that we would hope would be disease drivers. So the single most best characterized, highest confidence gene that is a driver of NASH is a gene called PNPLA3, where there's a particular variant that is known to be disease-causing. And so the first thing we did was we tested association between PNPLA3 and the pathologist scores. And we found nothing, 0. And then we said, well, maybe it's a power issue. So maybe our machine-learned version of the phenotype, maybe the one that's much more quantitative, that was trained towards pathologist scores but has much finer resolution, would be more powered to see that association. And the answer is marginally better, but still pretty much 0. What you see on the right-hand column in that left graph is what happens when you let go of any kind of anchoring on the pathologist scores. And you just say, let's look at the machine-learned, self-supervised embedding without any attempt to try and fit what the pathologist thinks is important. And voila, the single highest association of any gene in the genome, any variant in the genome, was PNPLA3 without a machine learning embedding. And you can see that that is a beautiful dose-responsive curve with a number of disease alleles. And so anyway, that demonstrates that the machine learning embedding is not only better powered, by looking orthogonally, you actually see things that are entirely orthogonal to what the pathologist knows to look at. Okay. I'm just going to show you these results, which are from the latest work that we've done most recently. This is now working on NAFLD, or now it's called MASLD, which is the actual fatty liver accumulation, which precedes NASH. And what we did here is an imputation from different types of modalities within the UK Biobank to, again, hugely increase the power of the number of patients for whom we were able to measure liver fat. If you just look at the number of patients for whom liver fat was measured, it's only about 4,600. With MRI, you can increase that, imputing from MRI, you can increase that to 25,000. Imputing from other molecular markers, metabolites or biomarkers, you can increase that considerably to up to 262,000. And so all of a sudden, you now are able to do genetic associations on a much larger number of patients than you were before. And this is what that looks like. For those of you who are not familiar, these are Manhattan plots. And this is, the top one is what you get when you just do MRI-derived liver fat, 25,000 samples. The bottom is what you get when you use the imputed liver fat, 262,000 samples. And you could also look at individual genes that are known to be disease-causing, and your p-values go way up. Now, I'm just going to show you this one result, which we haven't shown publicly before, so brand new, which is we took our favorite from the targets that emerged from this and put it into our cell-ML approach, which uses machine-learned phenotypes having to do with lipid droplet accumulation in hepatocytes. And we put the cells into this culture that has, the McDonald's diet for cells. And you can see that when you do that, they accumulate lots of green dots. The green dots is lipid droplets within the hepatocytes. And on the middle is our gene. And the right hand is DGAT2, which is a positive control. It's currently under clinical development by Pfizer for NAFLD. And you can see that the effect size of the reversion from our novel gene is actually comparable and arguably even slightly better. I would say comparable to what you get from DGAT2. So this is where the two lines of work converge. Okay, I added this section at Holly's request last night, knowing that I probably wouldn't get to it. So indeed, I did not, because I've been told that I have 5 minutes. But let's just say we have in order to take targets to molecules, we have a DNA-encoded library platform from which we collect very large amounts of binding affinity data and apply machine learning on that. We have an in-house DEL platform that is a second-generation DEL that I will not have a chance to go into, but basically has a number of different unique elements to it, including the ability to take the DNA barcode on one of those small molecules and use it to program small molecule synthesis rather than read out synthesis that was done. That allows you to do active learning because you can print DNA that targets specific molecules that you'd like to interrogate. We have a bivalent DNA-encoded library that hugely increases signal-to-noise. And we have a proprietary technology that basically allows you to rank order binders in ways that are much more high resolution than what you get from sequencing. All of that enables incredible machine learning to be done on these DNA-encoded library binding affinity data. And I wish I could show you these, but I'm not. All sorts of cool stuff where you can get improved performance. But in the interest of time and Q&A, since I'm told that I'm getting out of time, this is the platform that we've built. And it really is nice to have a platform that allows you to take all of these capabilities and just redeploy them across different problems. And as what we've seen is that things that used to take us 1.5 years 3 years ago can now be done in 3 months or even 3 weeks, or in some of our most recent work on oncology biomarkers, 3 days, because the more you deploy a platform, the smoother it gets, the better it gets, and the more insights it produces. And it's really nice to see that vision of pipeline through platform really manifesting and justifying the quite substantial investment that we made in building each of the components in this platform, as well as the integrated vision of it, how it all fits together. So with that, I will take questions. Thank you very much.
Yeah, thank you. That was incredible. So questions. Yeah. Yeah. Yeah, could you please come up to the mic?
Thank you for your inspiring talk. So you showed us many amazing applications of machine learning by integration of multi-omics data sets via embedding process. So my question is that, can we use machine learning again for embedding process like generative adversary network or reinforced learning to optimize the embedding?
So we've started to do some generative AI in various ways. So I showed you the generative AI for our GANs for visualization. All of the imputation is effectively generative AI, right? Because we're taking the embedding on histo and we're producing RNA-seq, we're producing attack-seq. So it is, in fact, a generative AI model sitting there in the background producing those data. And by effectively having this generative AI expansion of all of those into all of those data modalities, that is what powers the discovery that we have by associating genetics with RNA or attack with clinical outcomes and so on. So yeah, I mean, the embeddings really do serve a generative AI purpose as part of the discovery process.
Thank you.
Hi, Daphne. Made it here. I'm Fausto. I've just finished my PhD. Now we're co-founding a company. I'm a huge fan of Insitro. Really loved your talk.
Thank you.
My question for you is, sorry. So when it comes to novel target discovery, it's really exciting and really fantastic kind of new data sets that are coming out and new techniques to getting to this. The question that I have for you is in terms of, contextualizing those new targets, right? Because when you find something new, it's very fantastic. But usually you also want to contextualize it and really to find, I guess, the underlying root mechanism of the disease that you're trying to study in terms of that the novelness of your target. So I'd like to know what kind of techniques Insitro uses to better contextualize these new discoveries.
Yeah. So, I mean, first of all, I think it's important to understand the difference between two different goals, each of which is worthwhile, one of which is to understand and really sort of disentangle the underlying causal mechanisms of disease, and the other is to find a disease-modifying intervention. Yeah. And these are related, but they're not the same goal, and there's a lot of, drugs out there whose mechanism we don't understand for disease that we don't understand, and yet they work. And so I think what we're trying to solve for here is not necessarily the full-blown understanding of the disease, but really just disease-modifying treatments, where the thing that we focus on a lot is how do we gain conviction that a phenotype that we're seeing being modulated in our in vitro experiments or potentially in our in vivo embeddings, how well does that predict that this will actually modulate the disease in a patient? And that, I think, is really the fundamental question that we're looking to solve. Now, you do need to gain some mechanistic understanding of making sure that, it can't just be a random target, gene ORF number, blah, blah, blah. It needs to be something that you have some conviction around because it's a long and expensive journey from that point on. But you don't need to, we're not doing basic science. We're doing drug discovery.
Okay. Thank you so much.
Thank you for the wonderful talk. I'm here from University of Sydney, Australia. We are working on Parkinson's disease, and in Parkinson's, like about 10% is genetic and about 90% is idiopathic. For most of the neurological work that you spoke about, you touched on, you were playing on with the genes. Do you see a similar approach being effective for idiopathic Parkinson's as well?
I would not have had the confidence to say that up until recently, because the work on ALS was very much sort of in building mode without having had the chance to really see whether we see phenotypes. And you're never quite sure, especially when you have a degenerative disease that occurs in old age, whether you're going to see disease-relevant phenotypes in very young neurons that are like 10, 15 days old. And the answer is we see that in ALS. We see disease-relevant phenotypes in ALS, and we're able to screen for those, for reversion in those. Again, this is early days, and I don't want to say we have a target because it's very early, but at least we see phenotypes. So that gives me some hope that in other neurodegenerative diseases, we will also see phenotypes that are potentially relevant in neurodegeneration. I will say dopaminergic neurons are a bear. I mean, if you think motor neurons are hard, dopaminergic cholinergic neurons are harder in our experience, at least. So the path to get there is non-trivial, but I think there is reason to hope.
Sweet. Thank you.
Thank you. Going back to the kind of imputing attack data from these histology images, I was curious, since it sounded like your training set had like relatively limited number of cancer types. Is there evidence that it generalizes to new cancer types?
Oh, this was trained pan-cancer. So this was 23 cancer types, 400 patients across 23 cancer types. And we absolutely trained pan-cancer, because if you did not train pan-cancer, you were completely unpowered to do anything. In general, in our imputation work, which we've now done across multiple different modalities, you know, I showed you the ATAC-seq, I mentioned RNA-seq in the case of NASH. We've also done RNA-seq for cancer in the somatic alterations. You get so much power from training pan-cancer. So yeah, I would not advise doing it other ways.
Okay, thank you.
In the interest of time, we'll take one more question, and I hope you can network more during the lunch and coffee breaks.
Yeah, thank you, Daphne. This is Elvin. I'm from Cancer Dependency team. So we have a very similar project like you for the ATAC-seq. We have single cell share-seq for ATAC and the RNA in the same cell for 500 pan-cancer cell line. I'd like to know, did you ever have an open source package for your imputation study? Can we apply to our own data set here?
I don't know yet. This is fairly recent work. We haven't decided yet what we would want to release and at what stage. I mean, so maybe. We certainly have open sourced other things, including quite recently. So I don't want to say, so the answer is maybe. You're doing this in cancer lines, or you're doing
we
this?
are doing for 500 cancer cell line, including
Yeah, this uses
cancer.
ATAC. Sorry, this uses histo as input, not individual lines. And so we get so much information from the broader tumor architecture that I actually would be quite, I would say pleasantly surprised, but surprised if the same approach would apply to individual cells in a dish. The broader tumor architecture is quite important as a source of information. And a lot of the pathways that we see emerging in the ATAC imputation as being highly confident ATAC hits are things like cell-to-cell adhesion and various other, you know, tumor architecture things. So I mean, the technology would need, I mean, certainly the model would need to be at the very least retrained to have cell morphology, I guess, as input versus the histo, because it's quite different.
Yeah, thank you. Thank you.
So let's thank Daphne one more time.